think we should be ready for that. So everyone, I'm going to start the panel discussion on soil ecology, the microbiomes, and plant health. We have a very distinguished panel this evening, afternoon, morning, depending on where you're located. I'm going to start with Dr. Shashika. If you could please introduce yourself and who you are and where you're from, please.
Good morning and good afternoon to everyone. I am Shashika Hevavitarana. I represent Cal Poly State University in San Luis Obispo and Cal Poly Strawberry Center. And I conduct research specifically on strawberry, how to manage diseases of strawberry. Thank you.
Move on to Alberto.
Yeah, hello everyone. I'm Alberto. I'm the co-founder and the chief scientific officer at Biomakers. Biomakers is a Silicon Valley startup that is focused in soil health, that is using microbiome as a bioindicator at different levels, specialized to help farmers to understand what is the impact of their practices into the soil health, soil microbiome, the life in the soil, and how they can improve nutrient mobilization and also the healthiness of the different crops.
Thank you. Robin?
Yeah, hi you guys, I'm Robin Ross. I'm one of the co-founders for AgNexus. I represent the science and technology side, with my background being in plant pathology and microbiology. AgNexus helps companies with cool and interesting technologies, usually something really new and kind of industry changing, enter the market space.
Okay. Sarah, you're up next.
Hi, everybody. I'm Dr. Sarah Buddy Rodriguez. I'm the assistant plant pathologist at the BioSafe Systems, and I've been here for a couple of months. So I've been working on the regenerative, helping us develop the regenerative soils program at BioSafe Systems. And before this, I've worked with, I have a background in plant pathology, and I've worked with soil microbes afflicting potato for the past decade.
Great, move on to Kim.
Hi, I'm Kim Zinnick Anderson. I'm representing North Dakota State University. I am the lab manager and research specialist of the Soil Microbiome Lab. And our general mission in the lab is to study how different agricultural practices affect the soil microbiome and try to identify how to better work with the soils we already have for better crop productivity. My background is primarily in plant pathology with a little bit of soils in there as well.
Great. We're going to move on to our first question of the day. And this will be for each panelist as we go through. Please feel free to chime in as you feel necessary or appropriate. How do you evaluate microbial interactions? Dr. Shashika, would you like to start that?
Sure. As I mentioned, I specifically work on strawberry, but before that I worked on apple, specifically on soil-borne diseases. So my approach and interest was on how the soil microbiome interact in the rhizosphere, and this is beneficial in pathogenic microbial species. And I used microbiome analysis techniques to characterize the interactions of the plant and soil microbiome. This includes methods that involve molecular biology and also bioassays. So when we have a problem in terms of plant health, we know that there's a disease triangle where you have a pathogen and the host and the suitable environment, which would create a disease. And when we tease apart the interactions, we will have to focus on these interactions. And when we conduct soil treatments to manage the diseases, we evaluate the soil microbiome prior to application and after the application, and find out if that treatment significantly change the soil microbiome, especially in the rhizosphere. And currently the understanding is that rhizosphere is one of the most enriched niches of in the earth, and we use amplicon sequencing to characterize fungi and bacteria in the rhizosphere, and also techniques such as terminal restriction fragment length polymorphism, simply called TRFLP, to find out how these microbial groups are changing in response to the application of treatments. So once we see that microbial side of the story, we try to correlate that with the plant response, either with the production of fruit or the response to the pathogen challenges. So we artificially inoculate the soil to find out, okay, after treatment, what the plant response is. Can the plant accept that challenge and be resilient against the diseases? So that's what my approach has been.
Okay.
Thank you, Emily.
Okay. Does anybody have anything to add to that? Sarah or Dr. Kim or Alberto? And these are all your specialties.
Well, oh, I'm sorry, Alberto, were you going to say something? Okay. Well, from my previous research that I conducted as a postdoc, we kind of indirectly evaluated interactions between plant pathogenic organisms within complexes, because one of the things with soil-borne pathogens is that you never get a single pathogen. They always work in tandem with multiple other organisms to create what is referred to as these complexes. And while a lot of the technology that I used wasn't as sophisticated to get down to the very specific like chemical interactions between these organisms, one of the things that we did implement was the use of amplicon sequencing. So basically we went through and sequenced everything we could identify as a pathogen, and using a variety of different kind of statistics referred to as multivariate statistics, we were able to basically characterize and identify these communities. And from there, we looked at indicator species to kind of see which ones were the more abundant and frequently occurring organisms, and when they were in the presence and absence of other organisms within the communities, how that related to the disease severity and different kinds of crops. And so from there, we were able to kind of like develop some of these logistic regression models to kind of characterize what basically is going on based on the presence and absence of certain organisms. Now, currently, my new job, one of the things that we're hoping to delve more into is metagenomics, where we're actually sequencing a variety of different organisms, pathogenic or non-pathogenic, and basically, sequencing genes that have known functions or are related to different functions within certain cycles that are important in the soils, such as your carbon cycle. So, I think down the road, it's going to be a lot of like, well, if we take this organism out, what happens to the function of that particular community, and trying to relate that back. So That's a little bit more sophisticated. And if we really get into more of the newer technology, you can get into some pretty cool stuff. But for now, that's what we do, or what I've done.
Let's talk to Alberto about this. And obviously, you have the technology side of it. In how do you develop products to identify which microbes are beneficial?
Yes, as well, Kim or Shashika said, we are using amplicon sequencing to identify DNA in the soil, a specialized bacteria and fungal community, because it's the species that we have more information available thanks to the researchers that are working on that. Also, as Kim, we are using different approach. We are identifying the community of microbes that are living in that soils. A note, we detected today more than 5.5 different DNA chains in soils around the world. This is a huge information because only 500,000 of the species are known, and we know the specific taxonomic name. But also we are playing at functions. As Kim, we are identifying key functions in the soil and also predicting. From the taxons that we are identifying, it's possible to predict the total functions of that microbes. Another level that I'm implementing in our technology is the ecology. It's not only the microbes that are present, it's not only the functions that are present, it's only about how is the relation of that microbes. We are building meta-communities. It's like a technology similar like the Facebook or Twitter is using to predict social specific models in humans, but we apply the same technology to the microbes to understand how that microbes are working together in some specific place. That's perfect to understand at different levels how a microbial community is working. But one of the key questions, for example, for us, is about the disease risk or is about the nutrient mobilization. And the first is to understand if we have reference to know if something is low or something is high. In that case, we are building that reference. We have thousands of samples for at least 120 different crops around the world that allow us to understand if some specific function in some place in some crop is low, is because we have a reference of samples from that crop that have different values for the, for example, nitrogen mobilization. For us, it's key to understand what happened in the soil. No, but the approach is very similar. In the case of pathogens, for example, many people think, and I'm introducing here a discussion, many people think that sometimes it's about the abundance of the pathogen present in the soil, for example, that it can be infect the plant, and also, as Kim mentioned, the complexity of different pathogens that are living in the soil. But our data show, and we published it recently, for example, in vineyards, that it's not only about the abundance of the pathogens in a soil, because organic agriculture or biodynamic agriculture, we can find high abundance of pathogens. However, in conventional agriculture, we found that because of the application of fungicides specialized, decreased the abundance of pathogens, but that pathogens that are present in less abundance, like in organic agriculture, are more aggressive, or have more resistance to infect the plant. That's one of the key. For example, if we are thinking of what is the for example, horrible place or the place that we are infected and can kill us, a bacteria, for example, is a hospital, is the more cleaner place in the world. And the abundance of microbes in a hospital is very low because of the pressure of the cleaning. But the human pressure in the hospital to clean all the surface allow to identify bacteria that resist antibiotic, for example. In the same case in agriculture, place with an aggressive management and the conventional agriculture and their application of hard chemicals are reducing the pathogens, but that the pathogens are evolving to be more aggressive and to resist that treatments.
Great, thank you so much. So let's move on. Sarah, I'm going to ask you to continue on a little bit from what Alberto was talking about. Can you kind of explain the soil regeneration and the soil health that you're currently working on?
Sure. So it's still in development. We still, as I'm certain everybody on the panel can agree with, the soil, regardless of where you're at, next door, a couple, like even a lawn over, the ecology, the structure, everything is going to be different about it. So we're still in development of all of this, but our main goal is to try to figure out ways of knocking down some of those bad pathogens and then bringing back in some of the good. So a lot of the products that we use, such as hydrogen peroxide with peroxyacetic acid, can kind of help knock down a lot of those pathogens, but also some of the good bacteria and microbes that are in the soil. But then we've seen quite a bit of papers that discuss about the rebound of some of these microbiomes. So the good bacteria, and I believe from biomakers, we also we actually have some evidence that shows that the rebound of the good microbes are going a lot faster than that of the bad pathogens or bad pathogens. So a lot of what we're trying to figure out is finding that balance, especially within the different areas. Like the soil that you're going to see in Florida is not going to be the same as what you've got up here in North Dakota, or even over in Madrid, or in Cal Poly, or or in North Carolina. But what we're trying to see is that, yes, if we do knock all of these down and then we bring in some of those good microbes, such as like Bacillus subtilis, a couple of mycorrhizals, will that help benefit the plant, or at least help bring back and regenerate that soil microbiome, as opposed to just, forgive my phraseology on this, but nuking the soil with something that's as heavy as like a Medcam or some of the other more harsher chemicals that have been used in conventional agriculture for so long. But that's kind of the overall basis of what we're trying to do here, but we still have quite a bit to learn. But the preliminary data that we've gotten for the past year or two have been very promising.
Fantastic. Robin, you work in this quite a bit and with several different companies across the country and then some, I believe. What type of new technologies are you seeing with this and development of it?
Yeah, well, all kinds of new things as it pertains to the microbiome or biological types of products. I honestly think what Alberto was talking about is going to be a game changer, because your question of how do you measure the microbiome, or how do you measure changes in the microbiome, that's an awesome question that we never have been able to do in the past. And I think Biomakers is making a lot of headway there. One change, add a biocontrol agent or anything, it's almost like the butterfly effect. They could have a million downstream effects that we never did expect. So technology like Biomakers is going to be able to get their arms around that a bit. But there's other cool ones too. I kind of like metabolites as signaling molecules, and that's sort of an unknown thing now too. But what do they do? I'm sure they have many functions in signaling microbes and changing the microbiome potentially, as well as signaling plants. Microalgae, I think those are kind of cool. They have the potential to do all kinds of things, from working as signaling molecules, changing the microbiome, feeding other microbes, photosynthesizing and sequestering carbon. I think that one's going to be a game changer that we don't know a lot about, but I do like those. Here's just another cool one, bee vectoring technologies. The ones that's using bees to vector out microbes as biocontrol agents that deliver them directly to the flower where a potential disease might exist. So those are a bunch of cool new technologies.
Very cool. Thank you. Love hearing that type of information. I just got a question in from an audience member for attendee. Can you name a few bad pathogens and good microbes in the soil and the best way to mitigate impact of the bad pathogens? I'm going to open this up to anyone to answer.
So some of the research that we're that we're conducting right now is a rather large survey, and we're actually looking at mycorrhizal fungi, or arbusculal mycorrhizal fungi, otherwise known as AMF, and the colonization of such on the roots of spring wheat across the entire state of North Dakota. And there's been a lot of studies out there that would indicate that when you have an abundance or higher colonization of AMF fungi, that things like wilt caused by Fusarium oxysporum actually become, the disease severity caused by that is delimited, as well as sudden death syndrome, which is caused by Phytophthora. You know, the one thing now that people are starting to look more into is how things like tilling and nitrogen fertilizers actually impact that colonization rate. Because there's been a few studies that have been out there that would suggest that the higher the amounts of nitrogen fertilizers applied to your field, the lower the AMF colonization of your roots. Whether or not that directly correlates with the frequency of, you know, wilt in soybeans and sudden death syndrome, you know, they've alluded to it, you know, but of course, a lot of people are starting to try to delve more into what exactly that interaction is and how the different practices being implemented in the field are impacting that. So that's my 2 cents on those.
I would like to talk, I would like to answer that question using strawberry as an example. Currently in California, the major hindrances to strawberry production from soil-borne diseases are due to Fusarium oxysporum, Fomes specialis fragaria that causes Fusarium wilt, Macrophomina placiolana causing Macrophomina crown rot, and Verticillium dahliae that causes Verticillium wilt. There was a recent study by Lascano et al. That I also involved in to characterize the rhizosphere and bulk soil microbiome associated with certain cultivars of strawberries. And we conduct a cultivar trial to find out which cultivars and advanced selection lines that are resistant to these pathogens, specifically for Macrophomina and Verticillium. And we found that certain genera of Burkholderia and Pseudomonads are present in the resistant rhizosphere microbiome as opposed to the susceptible cultivars. So in that regard, we can see that in the core microbiome of a plant, there are specific microbial species that are associated with the genetics of that host, and we could further enhance the rhizosphere if we can help by amendment-based or microbial inoculant-based methods to further improve the health of the plant.
Yeah. Did anyone else want to add to that answer?
Well, for me, the question is tricky because, okay, we can mention 2 pathogens linked with a crop, but how many pathogens are in all crops, for example, are defined for all crops? We have a long list of pathogens linked with each disease per crop. And it's curious that there are some specific pathogens that are linked with one disease, but there are 100 pathogens linked with one specific disease. For example, in grapes, vines, Yesca, there are 200 different pathogens that we know that are linked with that problem, or Blackfoot disease linked with a problem in the roots. That's impressive But if we put in the context how many pathogens are compared with other microbes that we are able to detect, is less than 1%. Okay, and for me, the question is, how is the balance between pathogens, for example, a beneficial species, and what are the beneficial species, and how to know what are the goods and the bads, or how to classify that? In our case, we are using the network approach that is like a co-occurrence and co-exclusion. With a pathogen is in a co-exclusion with another species, that species have not survived in the same environment because one species is affecting to the other. And also we can detect species that need others, for example, to try to affect to the plant, no? To introduce themselves in the plant. Well, this is a community of microbes. And communities of microbes, we saw that in natural environments, they are in balance with each others. No, it's like all the microbes are together with others without problems, sharing functions, sharing chemicals, etc. But in the case of crops, humans is managing the system. Depending on the crop system, the interaction of the human with the environment is more hard or more less. In that case, we see that microbiome communities are specialized in the case, for example, of conventional agriculture. If you put nitrogen in the system, you are engaging specific microbes that like to eat nitrogen, and some of them can use the nitrogen to grow, some of them can use nitrogen to supply the nitrogen to the plant because are living in symbiosis or other things. Understand the balance of that communities when you are introducing something that can be a fertilizer, a crop protection product, etc., is the key. As we mentioned before, are necessary a lot of trials to understand control versus treated. What happened when you applied some specific product, for example. And for me, is the key. And also, when you are applying products into the system, because the microbiome, as we mentioned, is different in each location, because microbes, like animals or plants, evolve in a place in a different way. What we need to understand, if when you are putting a fertilizer, a crop protection, a biostimulant in one place, what is the effectiveness of that specific product under that specific community of microbes and environment, no? Physical-chemical conditions too. For me, is very, very interesting and is necessary, but it's not only about pathogens that are present of good or bad microbes, it's about how that microbes are in balance or not and in relation.
Alberto, I think you actually answered part of the next question that's coming in from the audience, but I'm also going to have this answered by our academia and Dr. Sarah and Dr. Kim and Dr. Shashika, because it's talking about, could changing soil microbes have unexpected consequences as it happens with more complex ecosystems? So since each of you are in different regions, I'd like to hear your comments on that as well. So Dr. Sarah, would you like to start?
Yep, yep, absolutely. So I was actually trying to figure out an analogy for this, because it's exactly what Alberto was saying. You need that balance. Even in organic systems, you can still find pathogens in those areas, but as long as they're getting outcompeted by everything else that's good in the system, you should not have any sort of a problem. So I kind of think of it like there's a bacteria that causes an ulcer in humans called Helicobacter pylori. It's always in your intestinal lining, but if let's say you eat something that's really high in sodium, or you decide that you really, really want to do that ghost pepper challenge and just eat as much capsaicin as you can, and it just throws off your system. Well, all that bacteria can really build up in your stomach and then outcompete all the good bacteria that's in your system and just overtake it and then cause ulcers. It's a very similar thing that you'd see in agriculture. If you're pumping in some different sort of fumigants or any other kinds of products, or just throwing a lot of fertilizer into your system, it may throw everything off balance, and then you may be causing more harm than good. So those, at least in our area, I know that could be, we're very big and Dr. Zitnik Anderson could also comment on this, but in North Dakota, we're a very big monoculture state. So a lot of our soil has been impacted in a very negative way over the past couple of decades. But yeah, I'll pass the baton on to someone else who wants to answer.
Dr. Kim, would you like to add to that?
Yeah, so I mean, yeah, Albert did a fantastic job of touching and a base on this, honestly. But yeah, I mean, anytime you introduce or you try to control an environment to your benefit, you are going to shift the paradigm that could potentially be detrimental in the long run. I think more than anything, we should probably kind of focus on trying to understand what is naturally occurring into our own specific regions. And I wouldn't say try to recreate that, because it's so complex. You're not gonna, I don't feel like you really can recreate that, but understanding and trying to identify those main practices that could potentially, you know, shift that balance. If anything, try to delimit the amount of destruction, I suppose, in the environment would probably be your best course of action. But one of the things that I've noticed is that when it comes to trying to understand what is naturally occurring in the environment, trying to get funding for that kind of research is difficult, because it doesn't like directly relate to the farmers. And so they're not going to see the benefit of that research like the following season, you know. So trying to get out and really see what's going on in the naturally occurring prairie native grasslands, it's kind of hard, because, you know, basically we're having to shoulder all of that, you know, the labor and the expenses that go into it. So I think more support for conducting those kinds of survey research it would be helpful, and maybe understanding, if anything, developing some basis of reference as to what we're doing to the soil and how it relates to what was there before could help us understand what we're doing to it.
Great. I think Robin wanted to add on to something with that.
You know, I would kind of go back to some basic plant pathology of the best fungicide is a healthy crop. So it's a complex system, as Alberto and Kim both just said. And I also want to tag in on that a lot of times we have gotten into kind of a chemical mindset where to control a pathogen, we want to do one thing. We traditionally have applied a pesticide. When you start thinking about microbes, I almost wonder if it's a better idea to start thinking consortia, because it's a very complex system, as you guys have been saying. And many of these microbes work in tandem with helpers and friends, and you're changing the entire system. And Kim, as you said, that does make funding difficult, because people like to fund a project or a product or something very finite. And these kinds of things are not so simple.
So I'm going to move on to, we got a couple more questions in here. What is the relationship between soil type and microbial balance, and how do crop yield goals fit into the profile? Alberto, you're smiling. Would you like to start that one?
Well, yeah, I have my specific thinking based on the data that I'm managing all the time. Well, there are two dimensions, right? Try to improve the sustainability in the soil, specialize after 30 years to degrade the soil. FAO data say that 30–40% of the arable soil was lost based on the hard application of agriculture system in terms of chemicals. This is something that needs to change, but we need to produce food and we need to maintain the fertility of our soils. For the other hand, is the productivity. No, we need more yield all the time to support the human needs. No, but for me, it's okay. We can recover soil. Maybe we are not be too much focus on productivity, but we need a balance between sustainability and productivity. Okay, and what is that in terms of microbiome? In terms of sustainability, network properties is an indicator. As much clusters of functions or more specialized is the system, less sustainability has the system. As less specialized, more nature environment we find is more resilient, but also maybe is not the most productive activity. In the middle, there is the term to engage sustainability, but also high productivity. And that's the perfect balance. In terms of societies of microbes, we have specific signatures that can support that idea, and we can measure that balance. Very simple. Like specialized needs are different clusters of microbes that are working together, but not all together at the same time. No, some of them are working in some direction, other in another direction. In the case of nature, all are working in the same way. Specialized is good, okay, in terms of productivity, in many different terms. Imagine society, imagine a company. If you are very specialized, you can be very productive, but not sustainable. Okay, that's my thinking. The balance is that try to be productive and be more sustainable. Our experience, for example, with testing different products around the world shows that sometimes an imbalance on biological application, for example, in the fungal community, reducing biodiversity, a term that is difficult to define, but biodiversity of fungal, then in some specific place, under some specific microbiome signatures and profiles, we saw a 40% of increase in yield, for example, in potatoes. That is huge, but only happen in some specific environments and specialized in low yield place. For my thinking is maybe we have a window or an opportunity to increase yield in low yield place, understanding the microbial communities there and also the chemical composition, okay? But this is a new opportunity. Try to increase the yield in high yield place. Maybe in high yield place, we need to be more sustainable than thinking more productive.
Thank you.
My opinion.
Did anyone else want to add to that answer? No? I think something that was brought up in one of our previous conversations, and I know a couple panelists were interested in it, it'd probably be interesting for the attendees to also know is, Roberto, can you talk a little bit on your research and the transparency that you have with having that public knowledge?
Sure, yeah. Well, in our case, that's how our, well, 40–50% of the people in the company is PhD in different fields. And we like to publish or to demonstrate the technology. Most of the panelists today are working with DNA sequencing as a tool to identify microbes. But then if we identify some feature, some specific biomarker, a species, a community, a kind of metric that we can lead with nutrition, disease risk, or sustainability, then we need to publish that. In our case, we know that each crop have a specific microbiome profile. Sometimes our knowledge can be applied in a crop-specific way. Sometimes we need to apply to every crop. But for that, we like to put the science of the markers that we are identifying in papers in order to specialize, pass the peer review. Other research can use our data to support our data to review our work, and we can demonstrate that something that we identify can work, and then scale that work, and also other research can try to replicate the same specific results in different crops. And it allows to improve the knowledge in a lot of directions, no? Because maybe I can be focused in some specific crops, but I don't know, Kim or Shakira are more focused in strawberries or in potatoes or in different crops, and sharing that knowledge is the best place or is the best thing. The last thing that I can mention is we have also a non-profit initiative that is calling Fields Forever, that we are partnership today with 160 different universities and companies around the world in order to improve the knowledge of the application of the microbiome in different fields. For example, one of our 7 topics right now in Fields Forever is how we can correlate carbon sequestration with microbiome signatures, no? Because we need a live biomarker that can change more quickly, for example, that the chemical approach.
Perfect. Robin, do you see a lot of the companies you're working with trying this educational and more open information to the farmers, the growers, the CEAs, whoever is utilizing?
No. I would say a lot of people are very closed with their proprietary technology. And so I really do admire biomakers for what they're doing there and getting that word out and publishing their findings. Although I will say that there's so many new technologies out there that collaborations to me are going to be key because you have the really big players and then you have all these new guys that have all kinds of new things. And without that kind of open collaboration, things are going to be a lot harder and some great technologies might not make it to the market.
Yeah. Dr. Shashika, would you like to add?
Yes. Being a representative from a public university, we always publish our work in peer-reviewed scientific journals. And prior to that, we will be disseminating that information. For example, we have an annual field day at Cal Poly Strawberry Center. We bring in over 250 grower and pest control advisor, industry representatives, and we disseminate that knowledge. Even if it is not the final product, we educate them the stages of our research, and then we disseminate our findings in scientific meetings like American Phytopathological Society meeting, and then finally we publish our work in peer-reviewed journals. So we have been 100% transparent about that, but we don't want to panic people with partial results. So we give some alert on, okay, this is not complete, but this is what we have so far, and then finally give the complete picture.
Yes.
Thanks.
Thank you. So, okay, we have one more question in from the attendees. If anyone else has questions from the audience, please feel free to put them in the Q&A. So the next question is, if you could please kindly comment on Pseudomonas perdita, I hope I pronounced that somewhat correct, as a PGPR, any experience on growth effects, and does it have an effect in fusarium control?
I would like to share some of the work that Dr. Mark Mazzola did on apple replant disease. And Pseudomonas perdita is one of the fluorescent pseudomonad species, and they produce bioactive compounds like antibiotics. And what Dr. Mazzola found was that certain apple rootstock genotypes that are susceptible to this soil-borne disease called replant disease harbor less of these species and more of other species that make that rootstock genotype more susceptible to pathogens like Rhizoctonia, Solani, Pythium, Phytophthora, and Pradolenchus penetrans, the lesion nematode. In that lab, we had research going on with Brassicaceae seed meal amendments to transform soil microbiome. And this species is one of the bacterial species that would increase in abundance after the applications of seed meal. And I know that this is used as a plant growth regulating rhizobacteria, but I haven't specifically worked on that or fusarium control. Maybe someone else has something to share about the fusarium control.
I would say that that's an interesting beneficial microbe. And one of the more interesting things about it is that it produces siderophores, which are strong iron chelators, which effectively work as a biocontrol agent by depriving the pathogen of available iron so that it can grow. That might be an interesting one to combine with other siderophore producing microbes, perhaps to even enhance its effectiveness even further. So I don't know if anybody knows too about the ability to propagate that biocontrol agent. I don't think it's a real easy one to grow and turn it into a commercial product.
Dr. Sarah, Dr. Kim, did you have any input on that? No. Okay, we got another.
I don't work with those. Sorry.
No worries.
It's out of my knowledge base, but I think Robin and Shashika did a great job answering that one.
Okay, moving on to the next question then from the attendees. Setting aside pesticidal activity of soil microbes, how important is biostimulating functions of these organisms, both for economic return in production, agriculture, and environmental stewardship and monocultural systems? And how can farmers better evaluate soil microbial activity, measure their populations and diversities, plus identify ways to connect species to functions to improve the fields? It's a long question, I know.
I'd like to tackle the first sentence. Somebody else has to get the rest.
The first one.
Okay, I'll go with the first sentence on that one. It's talking about biostimulant effects of many microbes. You guys that are in the Pacific Northwest right now know it is hot, hot, hot. It hasn't rained. California's in a drought. Arizona's facing water problems. And many of these microbes can improve tolerance to drought, improve tolerance to high heat, improve root growth. All of these, I think, are going to be key effects that these microbes can perform. So they are pretty critical. And right now it really displays that.
Thank you. Anyone else want to chime in on this?
Well, for us at Biomakers, for example, we try to summarize the information that are coming from the microbiome in 11 different indicators, divided in 3 levels. Biodiversity, to know the levels of biodiversity at the level of taxon, functional, resilience, that when they are applying the specific product, when farmers are applying products, then the disease risk and abiotic stress, for example, we are reporting microbes that are able to produce phytohormones, acid abscisic, phytokines, gibberellins. If you are adding a microbe that have that potential, depending on the level of the microbe that you are adding in the soil, that can be an effect or not to improving that specific phytohormone production. And then nutrient mobilization, this is another dimension that we are reporting, NPK and also mineral compounds. But to be very direct, imagine that you are taking a lion and you put the lion, I don't know, for example, in a Miami beach. Lion can survive or can die, okay, honestly. Sometimes it happens when you are putting a specific bioproducts in different places, specialized microbes. But for the other hand, we have a lot of different cases testing biologicals, and they have different effects. For example, recently we tested a Bacillus product in a tomato greenhouse place. And in this case, the product was very effective when they reduced the 20% the fertilization. In regular conditions, the product, okay, improve a bit the production, but when they reduce the fertilization, for one reason, Bacillus not only was working, if not changed a bit the community, specific improve some microbes, that was able to improve the level of potentially phytohormones that they can produce for the plant. And we report microbiome change, we report physiological change on the tomato plant, and also we test that the size of the tomatoes and the quality of the tomatoes was improving, including with less 20% of fertility. That was amazing. For me, was one specific case that was very interesting, but the same application in other crop, it doesn't work. Now that's sometimes works, sometimes not, depending of the environment that you are putting that specific product. No, that's the key. But some people think that, okay, biostimulants can improve nutrient mobilization, for example, nitrogen, phosphorus, potassium. Sometimes yes, but other times the effect can be to improve the level of phytohormone production when the plant is interacting with the microbes and specialized with the microbes that you are putting in the environment.
Yeah. So we have about 7 minutes left. This is what I call, we have one question in here. So actually that is for just us. So what I like to do is do kind of a quick round robin. Everybody gets 2 minutes to give what you want the attendees to have as a takeaway on the soil ecology, microbiomes, plant health. So Dr. Shashika, can you start off for us? Sure.
I think the understanding of soil microbiome has been more and more increased in the past decade or so. It was a black box. What's underneath the soil was a black box before, but with these newer technologies, we are able to find out more about what is the diversity, what is the functionality of the microbiome, specifically in specific niches like the rhizosphere or the phyllosphere. So there's a lot of literature that's available, that's been published, but I think what needs to happen is digesting that information and make it available to the growers who actually do the work on the ground. Because if there's a gap between the work that we are doing in the lab or in the greenhouse and what's actually happening out in the field, then the transfer of this knowledge is not going to be efficient. So I think panel discussions like this will really help to disseminate that knowledge. So I thank BioSafe for this opportunity to talk about microbiome work. Thank you very much.
Thank you. Alberto, what would you like everyone to take away from today's panel discussion?
Well, for me, I take some conclusions. First of all, microbiome is the second genome of the plant. We need to understand not only the microbes that are living there or identifying, if not the functions and the ecology. Second, yes, it's a new opportunity in many directions. We need to integrate the research, data, publication, etc., and decoding that data into useful information that can be accessed in a very easy way for farmers in a specific indicators. That's the work that Biomaker is doing right now. and also try to move in 3 different dimensions, no? Sustainability, disease risk, no? Reduce disease risks, and also improve the nutritional cycles in the soil to improve the plant growth. And as the audience say, it's necessary to increase productivity, yes, but in a more sustainable way. For me is the key.
Thank you. Robin, your turn. You're on mute, Robin.
All right, I was having a little bit of internet trouble. I couldn't hear you. I guess, and I better get this out in 1 minute because I think my internet's not too stable. The thing I would like as a takeaway is that I think we need to really get our mind around how the microbiome is a whole complex, big system. We've always thought of agricultural products as one thing, and we've continued to think about that even when it comes to microbes. If I could only find the right biocontrol agent or the right phosphorus solubilizing bacteria or the right nitrogen fixer, I think it's ever so more complex than that. And I truly admire Shashika and Kim and Sarah and Alberto for trying to get their head around this very complex system. And I think we will. They will eventually figure out how to manipulate the system and optimize the system.
Dr. Sarah.
Yeah, I think you hit that right on the head, Robin. Like, this is such a complex system, and the fact that we've got great academics like Shashika and Kim doing all the legwork trying to get everything figured out for us, and then people such as Robin and Alberto trying to figure out more of like the how is this interacting, what more can we get from this. This is basically our version of the space exploration.
Yeah.
We have it right at the bottom of our feet, and we truly are just scratching the surface on the knowledge of what is going on down there. We see what yields from it, but how it's making all of that magic work, we're not entirely sure. But what the biggest takeaway that we can see from this is that this is extraordinarily complex, but we know that they are the soil microbiome and the plant health and everything that has to do up above the ground are so interwoven, and we're just trying our best to not quite tease it apart, but understand why these connections exist the way that they do.
Right. Last but not least, Dr. Kim, what would you like people to take away from today's session?
I'm not sure what else to add after everyone's talked, but essentially, one of my old advisors used to say something to me that I tell the growers I work with, and it drives them bananas, but when addressing issues with soils, the soil microbiome, ultimately the answer is it's complex and it depends. Ultimately, it's the take-home message. We're working with a very unknown new frontier. We know more about oceans than we do about our own soils, and it's a very delicate system. So is there going to be a silver bullet answer to any one of these problems or questions? The answer is no, not really, because it's going to change with the environment. It's going to change with the region. It's going to change with the demands that each group of growers has in each region. So as we're trudging forward with some of this research, I guess my biggest thing is just be patient, but we need to keep that line of communication open between researchers and the growers. And I think people like Alberto, with the transparency and the level of outreach that the BioSafe company is doing right now, is something that's absolutely critical, because as Shashika pointed out, the growers are the ones who are doing all the legwork. We're just benefiting from all of their results at the end of the season. So we just need to make sure that we maintain this open line of communication, because we all need each other. So.
On that note, I want to thank all of my panelists for attending today. I do have a couple questions that came in, so I may forward them on for you guys to answer if you don't mind. And that concludes our panel discussion. Thank you all for attending.